And our next speaker doesn't really need much of an introduction. This is Jennifer Panagoulias, who leads regulatory and policy as well as compliance at FAST. And she also is the Chief Operating Officer at Transformatics Biotherapeutics. It's a real pleasure to welcome her. She's going to speak to us today. We're going to move to the clinical side of Pillar 4. And she's going to speak to us today about how endpoints are used in drug approvals. And I think it's really important that families understand this, because this is where your input at this stage is so critical, and for you to understand why all of this work and everything we're asking you to do and get involved with matters, and then how it's utilized and interpreted. So I'm very excited to hear her talk, and I encourage you all to pay very close attention, because this is where we're going to ask you to participate.
Thanks, Allison. Okay. All right, here we go. I'm Jennifer Panagoulias, co-director of the ABOM with Allison. These are my disclosures, which you nicely covered for me, so I'll just jump ahead. So I'm going to talk about endpoints and how they get used in the approval of a drug. And I'm going to start with some background, but part of why I wanted to talk about this is because I know with the discontinuation of one of the clinical development programs this year, there was a lot of question about, was it the endpoint? Do we have bad endpoints? Do we need more endpoints in studies of Angelman syndrome? And this is a lot of the work that we do at the ABOM, so I wanted to cover that and give you some background that could be helpful in reviewing studies on clinicaltrials.gov when you see things posted. So we'll start with definitions. What is an endpoint? And essentially it's a targeted outcome that gets analyzed to show that the drug is effective or that it's safe. So it's targeted outcome, but what is the outcome? The outcome is basically what you're looking to change. So you're trying to show a change in symptoms of the disorder. And when we think about Angelman syndrome, we would have relevant outcomes such as communication, sleep, cognition, seizures. So it's going to be something relevant to the disorder that you're studying. We use outcome measures to assess those outcomes, right? So these are tools that we use to measure the change, and ideally it should capture what matters most to caregivers or to patients with the disorder. And that seems obvious, but sometimes you'd be surprised that that gets missed along the way. So we do a lot of work to ensure that when we design a study, that we're including aspects of the disorder that are most relevant to patients and caregivers. And ideally, if you look at the bottom half of the slide, as FDA would say, the tool has to be appropriate. It needs to capture what you're trying to measure. So for example, if we were going to study changes in communication in an individual with Angelman syndrome, we wouldn't read a sentence and say, okay, now repeat this back to me so I can see how you communicate. That obviously would not be appropriate. So we need to make sure that we have measures that are able to actually capture the change. So we use things like the ORCA that allow us to assess changes in communication for individuals who are nonverbal and might communicate through gestures or AAC. And it needs to be able to be clearly captured in the labeling of the drug. And if you've ever gotten a prescription that comes in its original container, like a box, and you pull out maybe a bottle of pills and there's a piece of paper in there that's all folded up, that's the product's labeling. And that tells you about what they studied in the trials of the drug and about its safety and effects and other information like how to use it. So what matters most to caregivers of individuals living with Angelman syndrome are what's shown here. We actually go through efforts to research what is important, and we can do this in a number of ways. We might conduct a survey, which is shown on the right, which was a FAST survey, or a disease concept model, which is a qualitative research effort that gets put together through interviews of caregivers and clinicians. And this is from a paper that Terry Jo was involved in, where they looked at what's important. And you can see in both instances, really the top symptoms that are important that people would want to see changed from a treatment are communication, motor function, seizures, and some other things. So we make sure to incorporate that in studies. So the ABOM is a consortium, and I'm just going to kind of jump ahead here, which involves ASF and FAST and academics and all the industry members that are here today. And we get together and talk about what are the best tools that we have, and we want to make sure we have a whole slew of tools that are at the ready to be used in clinical trials. And what I can tell you is we've made a lot of progress here in looking at the symptoms that are important, that we should be measuring, and then making sure we have fit-for-purpose tools to measure them. And so what I want to leave you with is we have tools. We will continue to work and add tools to the toolkit, but at present, we have a lot of options that can be used in clinical trials to measure change. So now, how do you use them in the approval of a drug? So I'm going to give you an experience from a trial in Rett syndrome. And this is from a report. This is a report from a meeting of caregivers to share their experiences living with children that have Rett syndrome and what is important to them. And I want to highlight that tomorrow, Ryan Fisher is going to talk about patient-focused drug development initiatives more, and that's going to be an amazing talk, so don't miss it. But from this report, one of the things that came out of it was a need for change in aggressive behaviors. And what we saw from the community here was they said, girls living with Rett syndrome they can have breathing difficulties, they can have emotional and behavioral problems. And this is important. What families wanted to see changes there because it was really disruptive. It was ranked as an important symptom among many others. And now this is a posting of the Lavender study, which was a phase 3 clinical trial in women with Rett syndrome. And this is what was measured as their primary outcome. So I'm going to step you through this now. So you could go to clinicaltrials.gov and look at study designs for Angelman syndrome and see information that's laid out in the same format. And we'll start at the top here, which is what is the study measuring? So if you go to that box, this will tell you what are the outcomes of the study. And we can see here that the outcome of interest is behavior. And we know it's meaningful from the prior page in the report from the community. And the outcome measure to look at behavior is the Rett Syndrome Behavioral Questionnaire. And then the endpoint, here we go, is the change in behavior on this scale after 12 weeks of treatment. So that's the actual endpoint. The endpoint is what happens on this scale after treatment in the treated group versus placebo at 12 weeks. And another thing we learned is this is the primary outcome measure. And so when we design a study that's going to be providing evidence of the drug's effect and safety, we pre-specify what we're looking at. And we rank it, and that means what we're going to test in what order. This study actually had 2 primary endpoints, or co-primary endpoints, but it tells you this is the primary, so this is first. I'm going to look at this first when I analyze my results. And what they saw at the end of the study was that the group in purple, which is the treated group, had a larger reduction in their behavioral score on the Rett Behavioral Questionnaire than the placebo group in gray. And in the bottom panel, on the other co-primary endpoint, which was the impression of improvement as rated by a doctor, that the purple group had more improvement than the gray group. So the treated group did better than the placebo, and the study met its co-primary endpoints. And what happened? The drug got approved. And this is a snapshot of FDA's page where they made an announcement, hey, we approved the first treatment for Rett syndrome. And in the box, this is from the labeling of the drug, you can see they explain the company that ran the study looked at the Rett Syndrome Behavioral Questionnaire. And this is a scale that measures behavior. It's scored from 0 to 2, up to a score of 90 points. A lower score is better. And it goes on to say that the drug lowered the score for the treated group versus the placebo. And so it measures what was important as assessed by caregivers. It was able to capture the concept. It was a scale for Rett, not a scale for Alzheimer's. And it can clearly be described. And that's how endpoints get translated into labeling. And FDA said this provided us clear data that supported approval, and we thought it was a meaningful benefit, so we approved the drug. So now just a word on biomarkers. That's the B in the ABOM. That's something that we focus on. So biomarkers are not clinical outcomes. So they don't measure how a person feels or functions, but they're usually something, a characteristic that's objective. So it's a blood test or an MRI or an EEG in Angelman syndrome. And what we're focused on, because there's many types of biomarkers, are those that show the effect of a drug or that might serve as an endpoint. So biomarkers are important in a lot of ways. They are objective. So the examples that I just gave you of outcome measures, which measure how you feel or function, some of them are subjective, meaning that it's really in the eye of the beholder, the rater. So if you have a measure that includes a rating, I'm going to rate you on a scale of 1 to 7. The way each doctor makes that rating might be a little bit different, and so there can be subjectivity. Or if it's rated by a caregiver, you have thoughts about how your child might be performing, and it could be sort of biased by certain feelings that you have. So it's more subjective. Whereas lab tests are clear, it's a number or it's a picture, and so it's objective. So biomarkers are important because they help understand the effects of the drug in a more objective way, and they can help bolster up results that are subjective on clinical measures. And biomarkers can be really important because when you have good ones that you understand, they can revolutionize the development landscape. And what I mean here, I'm looking at an example from the field of multiple sclerosis where advances in MRI technology help them understand that if we see activity on the MRI in the brain of patients with MS, we know that's a bad marker for what's going to happen clinically. They're going to have more relapses, more activity that manifests neurologically. And they did a lot of studying to know that that relationship was a pretty tight correlation. And so they could see that if we use this MRI, we can tell in about 3 months' time if a drug is working or not working. And what that did was really bring a lot of treatments into the space because you didn't have to spend a lot of money doing super long studies to see that your drug didn't work. It's like, okay, this is working. We can see in 3 months that we have an effect. So let's advance this program deeper into development. And these aren't even all the drugs that are approved in MS. These are just some that I could fit on the slide. So really it helps open the door to be able to look at things more quickly. The other opportunity that it gives us is one to think about using these biomarkers as endpoints in a study. So what you're going to use to base the approval on. And they're called surrogate endpoints because they're not a clinical outcome measure that looks at how you feel or how you function, but it's a lab value usually. So it's a biomarker. And it's thought to predict clinical benefit because it's not showing, again, changes in functioning. It's showing changes maybe in a lab value. And a classic example of how this gets used is in trials of stroke. They often look at blood pressure. So they know from many, many studies that if you lower blood pressure, it's going to lower your risk of stroke. And they can look at blood pressure because they may need to follow patients for 10 or 12 years to see that the stroke risk goes down. They're waiting for people to have or not have strokes. So sometimes you use biomarkers as surrogate endpoints when it takes a long time to read out the clinical measure. But you have to accumulate a lot of evidence. I don't want to make this sound easy, like, oh, we have a biomarker, now we can use it as a surrogate endpoint. It's harder than that. You have to understand, what does the biomarker mean to the disease? How does it change over time? Can I move it with treatment? Does it go up? Does it go down? How does it relate? How does it predict what will happen with clinical outcomes? So we do a lot of this work in the ABOM. And one thing that has sort of risen to get more visibility is delta power as a biomarker in Angelman syndrome. And this is an aspect of the EEG that's very characteristic in Angelman syndrome. And if you look at panel F, all you really need to take away is that individuals with Angelman syndrome are in the red bars, and neurotypical individuals are in the black bars. And you can see the red is always increased. And we also see this in the mouse. So this is great. In the mouse model of Angelman syndrome, they have that same delta power feature. So we can study it. We can look in animal models to understand if we're moving that with treatments. There we go. We know that delta power decreases with age in Angelman syndrome. If you look at the top panel and the red line over the gray, you can see that there's a decrease with age. As patients age, their delta power goes down, but the red is still always above the gray. So patients with Angelman syndrome still always have higher delta power than neurotypical individuals. And we know that patients with a deletion have a little bit higher delta power than those with non-deletion mutations, but still both groups are increased over neurotypical. So it's a pretty strong marker. So then we start thinking, okay, well, what do we know about delta power and how might it have a relationship to clinical measures? And this has been really amazing work that's been done by some of the industry groups that are involved in the ABOM to show that there's a relationship between delta power and cognition. And what we see is the higher the delta power, the lower the predicted cognitive score on the Bayley scale. So there's a relationship that gives us something to look further at in terms of if we move delta power and we moved it down more into the neurotypical range or lowered it, where would you expect to see the change clinically? And importantly, there's been some work done in mouse to show that if you give the mouse a mouse ASO, this is a mouse-specific ASO that targets the UBE3A antisense transcript that you heard about this morning in Allison's talk, you can lower it with treatment. So if you look at the middle group, in blue, which is the treated mouse, versus the green, which is a neurotypical or wild-type mouse, you can see that the delta power is coming down over 8 weeks in this study. So with treatment, you can lower it. So you can move the marker with treatment, which is important because you have to be able to move it to show an effect. So we get really excited about the premise of surrogate endpoints because they can really change the development trajectory. You can shorten the overall development time. We had a great presentation from our statistical team yesterday that showed us how biomarkers can help you shorten the development life cycle because you can look at them faster maybe than you can look at clinical outcomes if they move and change earlier. And people always ask me, has this ever been used? Are there approvals in rare disease on surrogate endpoints? And there are. There's 4 oligonucleotides in the space of Duchenne muscular dystrophy that have been approved on the basis of a surrogate endpoint. And FDA put out a guidance where they said, we've learned so much, there's been so much data amassed here where we understand if you lower this, or you, in this case, you increase protein levels in patients with Duchenne muscular dystrophy, you could expect to probably see changes clinically, and because we've learned about what that biomarker means within that disorder. So it's really important. It doesn't mean that you don't confirm clinical benefit. You still have to look at that. And so when a drug is approved on the, just want to back up one, on the basis of a surrogate endpoint, if you look in this box, all you need to see is the last sentence, which says, yeah, we approve this drug, but we have to confirm clinical benefit. And continued approval is really conditioned upon showing that that biomarker did predict that clinical changes would come later. So those are important studies that have to be done and finished. And it's a commitment, it's a formal commitment, and FDA has regulatory enforcement power over certain commitment studies to make sure that they get done and do show that the drug provides some clinical benefit to patients. This slide is busy, but what I want to walk you through here is this is a recent example of regulatory flexibility around surrogate endpoints using a biomarker and why they're so important to bolster up data. So if you look at the first box on the upper left-hand corner, you can see that this is a drug, tofersen, which was recently approved for a very small subgroup of genetic ALS. And the study was using a ALS-specific rating scale as the primary outcome measure. And it did not meet its primary endpoint. It failed that primary endpoint. And so in bold, these are quotes that I took from FDA's review package for tofersen, where it said, well, the study didn't meet its primary endpoint. But in the box below, we did see that there was a change in this biomarker called NFL, a blood biomarker that was really a marker of neurodegeneration of cells, neurons dying. And we know that that's bad, and we know that the more that happens, it predicts progression and overall survival in ALS. There's been so much research done to show that. And we saw that there were patients whose NFL levels were going down, and in those same patients, some of the ones with the biggest decreases in NFL were having changes in their rating, their functional rating of ALS. So we think that it was moving in the right direction, even though the study didn't meet its primary endpoint. And when they looked at the total data package overall, they decided to exercise regulatory flexibility and say, you know what, we're going to still approve this using accelerated approval, meaning the company has to confirm that it will show a clinical benefit in the long term, because we think it makes sense for patients, and we want to give the patients a chance to have this drug and to derive some benefit because this disease has such a massive unmet medical need. And that's really what we're looking for. We're looking for some regulatory flexibility in these settings where they understand the unmet needs of patients. And so I will end there with a big thank you, because it's your involvement in natural history studies globally that helps us understand outcome measures and biomarkers. And we learned so much of the natural history study data is focused upon us understanding these outcome measures and how they change or don't change over time in individuals with Angelman syndrome, and how biomarkers change or don't change. And so it gives us this toolkit to work with for clinical trials. Thank you.